So for part 2 of chemical principles, we're going to focus mostly in on the organic molecules. All right, so organic molecules are going to contain carbon bonded with hydrogen. All right, so carbon and hydrogen are going to be together here and they're going to be bonded. Now this bonding is going to be through covalent bonds. This allows the molecules to become quite large. So most of the time they get termed as macromolecules, macro meaning big or large. They normally have a carbon backbone. Okay, so we have a carbon backbone with functional groups that are attached to it. The functional groups are the parts that actually react with other molecules. Some examples of some functional groups are shown here, things like carboxyl, aminos, or phosphates. These are going to be some of those that are tagged on to that R chain, which is the carbon backbone, and this changes up the properties of that particular molecule. Some main examples that we see when we're talking about organic molecules are carbohydrates, lipids, which are fats, proteins, and nucleic acids. And you can actually see here that carboxyl groups a lot of times are found in lipids and proteins. Amino groups are found in proteins, and we also see phosphate groups are going to be found in nucleic acids and ATP. So these are just showing you some of these functional groups and what kind of molecules they are going to be associated with. What we're going to do in this particular lecture is we're going to look at each of these macromolecules in a little more detail, and we're going to start with the carbohydrates. So guys, carbohydrates, when we talk about their typical structure, they're going to have 1 carbon to 2 hydrogens to 1 oxygen. So there's going to be a ratio here. There's a 1-to-1 ratio. Okay, so we have CHO, but it has a ratio. This is going to be where there are double the amount of hydrogens than there are carbon and oxygens. A lot of times when we represent this, they put it in parentheses with a little n. This n equals the number of carbons that are present. Now, when we talk about carbohydrates, they do have a variety of functions. So when we look first at monosaccharides, these are the simplest sugars. They have anywhere between 3 to 7 carbons. One of the most common are what we call the hexose sugars. These are glucose and fructose, and hexose tells you that they have 6 carbons. They are going to be your immediate source of energy, and they're the immediate source of energy for majority of the organisms. Pentoses are going to have more of a pentagon type shape, and a lot of times they're going to have 5 carbons to them. This is ribose and deoxyribose, which are important sugar bases to the formation of RNA and DNA. Now, we can go from a monosaccharide and we can put 2 monosaccharides together, and when 2 of them come together, it's called a disaccharide. This is a monosaccharide plus a monosaccharide. Some common disaccharides are things like sucrose. Sucrose is how your plants are going to transport a sugar that they make through photosynthesis. And in animals, we see it mostly as lactose. Lactose, guys, is the type of sugar you find mostly in milks. Now, when you add even more monosaccharides to that structure, then it becomes polysaccharides. Polysaccharides are going to be up to thousands of monosaccharides that are put together. Some examples of these are glycogen, cellulose, starch, and what we call chitin or chitin. So guys, glycogen is how animals like ourselves store our excessive sugars, like in our muscles and our liver. Cellulose is the structural sugar that plants use, okay, to help be able to go up against gravity. It makes up their cell walls mostly. Starch is how plants store their extra sugars. That's why potatoes have so much starch. And then chitin or chitin is going to be in the cell wall of fungus as well as in the exoskeleton of most insects or bugs. Now, on the microbiology side, some microbes can produce enzymes that actually can hydrolyze disaccharides and polysaccharides back into monosaccharides for energy. Okay, some don't. Some of them have to rely on monosaccharides alone for energy, but some have special enzymes that allow them to break down more complex sugars into simple sugars for energy sources. Also, disaccharides and polysaccharides are sometimes used by microorganisms for structural components, okay, to help give them structure or support. So here's just showing you kind of an example. We see those monosaccharides. Monosaccharides have the pentose, and you can see that they have a pentagon shape. We then have the hexose. Now, this middle one, fructose, doesn't look like a hexose sugar because it still has a pentagon shape, but it does have 6 carbons present. Since it has the 6 carbons present, that's why it's called a hexose sugar. When we put 2 of these sugars together, we get a disaccharide. So this top picture is showing you lactose as a disaccharide. And when we have a bunch of these sugars put together, we have a polysaccharide. All right, so now let's look at lipids. Lipids are fats. They are going to be insoluble in water. They do not like water, so they are considered hydrophobic. They are soluble, however, in alcohol. So alcohol does dissolve away lipids or fats. When we talk about simple lipids, they're known as fats, and they're composed of 3 fatty acids plus a glycerol. These a lot of times get termed as triglycerides because you have the 3 fatty acids on the glycerol for the triglycerides. Now, triglycerides can be either saturated or unsaturated. A saturated triglyceride is going to be where it is saturated and completely covered with hydrogens. See, all the carbons are going to be covered in hydrogens in these first 2 chains. These are known as saturated fats. On the other hand, if they are not completely saturated with hydrogens and they contain a double bond between the carbons like you see in the last chain, that's called an unsaturated fat. There's some spaces missing from hydrogens, and so it's unsaturated. A more complex type of fat is known as a phospholipid. Phospholipids are going to make up the plasma membrane of living cells. These plasma membranes with these phospholipids are going to have a very unique structure because a phospholipid is hydrophobic on its tail side with the fats, okay, but it is hydrophilic on the head side, at the phosphate area. So this is going to allow it to create that barrier you see there where the tails are pointed together and there's water outside, but there's no water in the middle between those tails. Another complex type of fat that we do see here in microbiology is what we call the mycolic acid. These are going to be in the cell wall of some types of bacteria called the mycobacteria. This mycolic acid is having more complex structure than we would see with just using sugars in that cell wall. Another type of fat are steroids. Steroids are things like cholesterol or sterols, and they're embedded within the plasma membrane. These are actually going to help the plasma membrane have a little more fluidity to them. And in the plasma membrane of animals and also the mycoplasm along with fungi, we do find these particular steroids. They're fused carbon rings. If you'll notice, steroids kind of look like honeycomb type structures. All right, the next macromolecule we want to discuss are proteins. Proteins are going to be built out of amino acids. Amino acids are the building block of the protein. Now, when we look at an amino acid, the amino acid is going to have an amino group on one side with that NH2, and it's going to have a carboxyl group on the other side. The carboxyl group gives it that acid type of structure. So this is where the name amino acid comes from. These 2 groups are bonded to a carbon that's in the center, and then there's always going to be an R group that comes off, and this R group is the remainder of the molecule chain, and that's going to determine what amino acid you're looking at because there are 20 different types of amino acids, and the part that's different is the R group, known as kind of like the remainder or reference group. So here are the 20 different amino acids. They do get named by that R group that's present, and the R groups are highlighted for you. Now again, I'm not going to ask you to memorize the 20 different amino acids, but I do want you to understand that they are different based on that R group. All of them have the amino group and the carboxyl group that are located on that central carbon. Now, these 20 different amino acids are going to be helpful in the whole formation of proteins. So amino acids, when they start bonding together, they're going to form, of course, dipeptides first, then polypeptides, then proteins. So when they start forming these polypeptide structures, they start to look like a string of pearls. The string is the peptide bond, and the little pearls are the different amino acids. However, in order for a polypeptide to become a protein, there's going to be some special folding that's going to need to take place. So a concept here, when we're looking at proteins or polypeptides, they have a variety of structures and functions. Some of these are going to become enzymes, others are going to be exotoxins, they could be structural components, and the list could go on and on. They can play lots of different roles depending on what combination of amino acids we put together. Now, in order for the protein to become functional, it does have to undergo some folding in a specific way. The primary stage of a protein is just that string of pearl stage. It's a polypeptide chain, a bunch of amino acids joined together. It's not functional here. We then see it goes into the secondary structure, and in the secondary structure we see that the string of pearls is going to start to have some alpha helixes, where they're going to start to go around kind of like a spring, or they're going to start to form beta pleats. Okay, so kind of like a pleat in a skirt or something like that, or if you were to take a piece of paper and you make a fan out of it, that's how those pleats look. Again, though, in the secondary structure, this protein is still not functional. In the tertiary structure, we see that it's going to have more of a 3D form. And guys, this is known as the globular structure. This is where it looks like we just took those folds and those twists and we just mingled them together and wadded them together. That's kind of what they did. This gives it its 3D shape. Some proteins are functional in this tertiary shape. Other proteins have to get to the fourth stage, which is the quaternary stage, in order to be functional. This contains more than one type of polypeptide. So you'll notice there's a light blue polypeptide that's going to be bound to a dark blue polypeptide chain. And so these two working together make that functional protein. Okay, so some can be functional at the tertiary, others have to be functional in the quaternary stage. Now, this folding is so important because if a protein unfolds, it no longer works. And when a protein unfolds, we say that the protein has been denatured. Denaturation is a change in a protein shape due to the breaking of bonds. This causes the protein to no longer work. It's non-functional. And if it's damaged enough, it could be permanent. We cannot make it go back to its normal folding. There's a number of things that actually can denature a protein. Temperature is one of them. If proteins get too hot, they can denature and they're no longer functional. Okay, pH is another one. This is one reason we talked last lecture about buffers and why we need to buffer pH changes, because if the pH changes too drastically, proteins can denature and they no longer work. And also salt concentrations. So these are all things that can actually cause the protein in its normal shape to unwind, and now it's no longer working. The last macromolecule we're going to look at are nucleic acids. Nucleic acids have a building block called the nucleotide. A nucleotide is going to be composed of a phosphate group. Okay, so we have a phosphate group there, a pentose sugar, so it's going to have this sugar base. It will either be deoxyribose or ribose, depending on which type of nucleic acid we're building. And it's going to have a nitrogenous base. Now, the nitrogenous base is going to either be represented by the letter C, A, G, T, or U, and these are different types of nitrogenous bases that can be present. Now, the A and G stand for adenine and guanine, and those are known as purine. They have a double structure where they are connected to a sugar. See how the double rings that are there? Pyridamines on the other side are the cytosine and the thymine, and they have a single ring structure. And one way to remember these guys is pyridamine has a Y, so does cytosine and thymine. Now, uracil is also part of this group because it takes the place of thymine when we are talking about RNA. Now, DNA structure was actually first pretty much discovered and put together by Watson and Crick in the 1950s. Well, they're at least accredited for it. They came up with the first kind of 3D model of what DNA really looked like. We notice that the DNA, there is complementary base pairing that takes place. A is always going to pair with T, and G is always going to pair with C. And again, another way to remember this is that the straight letters, the ones that have the straight sides, they pair together, and the curved letters pair together. Now, this is only in DNA because there's a pairing for a double helix structure. So hydrogen bonds are going to form between the two sugar phosphate strands, and this is what creates those rungs of the ladder. Deoxyribose is the sugar base when we're looking at DNA. So it is a double helix, which means it has two strands and it twists like a ladder, like a twisted ladder or twisted staircase. That's what gives DNA its special structure. On the other hand, RNA is a single strand. And when we look at RNA, it has a different sugar, which is called the ribose sugar. And it still has the base pairs of A, G, and C, but it does not have the T. T gets replaced with U, which is uracil. RNA is important because it's going to be used in order to make proteins. There are three types of RNA that are going to help make a protein, and they include mRNA, which is messenger RNA, and you can see that in the kind of the pink color. We also have tRNA, which is transfer. They're going to actually transfer the amino acids in order to build the protein. And then the rRNA, shown here in the brown, is what's going to make up the ribosome. Ribosomes are the structures inside of cells that actually produce proteins. They do the protein synthesis. Okay, so all three of these types of RNA have to be present and have to be working in order to create functional proteins. So here's a quick comparison of DNA and RNA. We see the strands are different. DNA is a double strand, RNA is single. The composition is also different. DNA has a deoxyribose sugar, whereas RNA has a ribose. The bases. DNA has A pairing with T and G with C, whereas RNA is going to contain A, U, C, and G. DNA's main function with heredity, it's going to store your genetic information, whereas RNA is associated with producing proteins. One last thing we need to talk about that doesn't really kind of fit into these macromolecules of a nucleic acid, a protein, things like that, is adenosine triphosphate, or ATP. It is a type of nucleotide. It is not a nucleic acid, but it is a type of nucleotide, and it has a special structure that you can see here. ATP is going to be composed of a nitrogen base, adenine, composed or bonded to a ribose sugar, and then it's going to be connected to three phosphate groups. That's the tri. The triphosphate tells you there are three phosphate groups present. Well, what's the function of this? Why are we talking about it? Well, ATP is important because its function is it's the principal carrier of energy. It's the currency of energy for cells. Okay, and so this ATP is how a cell is going to be able to utilize energy and be able to grow, reproduce, develop all those things it needs when energy is required. Now, if you'll notice, though, we see that there is a constant reaction that's going back and forth. So this is a reversible reaction. We see that we may start with ATP, adenosine triphosphate, but when we need energy to be let out, we're going to break that last bond, that high energy bond between the last phosphate. We're going to break it off, and that ends up causing it to be formed into adenosine diphosphate, plus the phosphate that was broken, and energy gets released. However, there are times we need to store energy. Well, this is where we're going to take energy and we're going to store it and push it with the last. This is where we're going to take energy and we're going to attach it with that phosphate, and we're going to go back from ADP to ATP. So there's a constant back and forth that's happening inside of a cell in the use of ATP and the storage of energy in forming ATP. So some concepts here. Some molecules may have a combination of different types of organic molecules. When we look at bacterial cell walls, gram-positive cell walls mostly contain a structure called peptidoglycan. Peptidoglycan are a bunch of disaccharides with proteins or polypeptides as cross bridges. That's what creates that peptidoglycan. We see that gram-negative cell walls have a thin layer of peptidoglycan, but then they have a thicker layer of what we call a lipopolysaccharide. This is what allows it to become more of a problem. It causes it to be more pathogenic and producing also some endotoxins. So these are going to cause some differences in different types of bacteria, and we'll talk more about a gram-positive and gram-negative a little bit later and what that means. Now, information from this particular presentation over chemistry is important because you're going to start to see concepts of it throughout this course. We see that you're going to be applying some of this stuff in the functional anatomy of cells, which will be discussed in chapter 4, microbial metabolism in chapter 5, growth in 6, and also control of growth in chapter 7. So these principles are important because they're going to help you build on concepts that you are going to see in the future chapters.